you, Rachel. Very important last slide. We'll let the PIs know that was included. All right, and our last speaker is Emma Hodcroft, who will be presenting her work on Pathoplexus. So please, Emma, or sorry, rather, she threw me off because she went on the other side. Please welcome Emma to the stage.
So it is so exciting and also a great honor to be here today at PhageCon to talk about Pathoplexus. Oh dear, getting ahead of myself. A new database for viral genomic sharing that you've heard a little bit about already, which is in particular a community-driven solution for transparent and equitable viral genomic data sharing. Now, I'm pretty sure I don't need to tell this audience about the importance of data sharing. In fact, we've had a whole panel covering it this morning. But, of course, data sharing is really critical to doing science. And just to give a few examples, it is, of course, sharing viral genomic data that lets us do things like identify new variants or new mutations, to see how a virus might be spreading globally or within communities, and, of course, to develop things like vaccines, antivirals, treatments, and effective public health interventions. But despite the fact that we know that viral data sharing is critical, we also know that there are real challenges there and that data doesn't always get shared. Now, there are many reasons for this, and some of them I'm sure we'll discuss in more detail at this conference. But two big reasons are the technical difficulty and concerns about credit. For example, if you are someone who's more lab-based, or maybe you're a clinician, or you're just in the middle of an outbreak or an epidemic, if sharing data is too burdensome, it won't be at the top of your priority list. You won't do it. You might do it later when things calm down or when you can get someone else to help you, but that doesn't mean that that data is available now when we need it to inform our response to an outbreak or to better understand a new pathogen. So we need to be honest about the fact that you shouldn't have to have a strong computational background just to do data sharing. It should be something that's easy enough to be accessible to everyone. And whether we like it or not, in academia and in research, publications are very much, sorry, my talk has a mind of its own. Publications are very much the currency of our livelihood. And so we need to be honest about the fact that if people feel like if they share their data, it's going to be scooped, that someone else will publish on it before they do, then again, they will not feel like they can share that data. And if that means that, of course, again, that data will be shared, it will get out there whenever the publication is ready, but we won't have it now, not when we might need it. And we may not like that incentive structure, but that's another talk. This is the incentives in the society, academic society we live in right now. Now, there are, of course, pre-existing viral sequence sharing options. I'm sure most of you have heard of them. One of them is INSDC that we already touched on today. What you may not know, which we did touch on a little bit, but maybe didn't explain so clearly, is that INSDC is actually a federation of more than just one database. So it comprises, at the moment, GenBank, DNA Databank of Japan, and ENA. And if you upload data, for example, to GenBank, it will appear on DDBJ and ENA as well, and vice versa. So all of that data is synced. There's also GISAID that was founded in 2008 in response to the swine flu outbreak, where there was a big issue with people sharing data or not sharing data. They provide some protections to data sharers, and that has really encouraged more data sharing. So providing that sense of security is really important. Now, both of these existing options definitely have many pros. You've heard of them. They're well-established systems. They contain a lot of data, and that's really important because we don't want to have to go to 15 places to try and put together a data set. They have trust in the community. People are used to using them. They know how to use them. And of course, they've been around for a while, so they will probably be around tomorrow. We have trust that they will continue existing. On the other hand, though, there definitely are some cons. For example, there's really no flexibility in licensing. If you put your data on INSDC, in GenBank or ENA, it is completely open. And apart from the kind of ethical side, anyone can take that data and publish on it. They don't need to get in touch with you. GISAID does provide some protections there. It cannot be used for everything. But if you ever want to change that, you need to re-upload it to INSDC. You can't change that license or have other options there. Getting data in and out can be another big barrier. If any of you have ever uploaded, for example, to GenBank, I've never met anyone yet that said that was just an amazing day that they really enjoyed. So that's a real barrier, right? If it's that difficult, it's not a big incentive to share. Getting data into GISAID is much easier, but for both of these, getting the data back out again can be a challenge, and that can be another hurdle that people run into further down the road. There's also limited interoperability, so you can't really move data between these two databases. You have to upload it twice, once to each of them. And then finally, even though both of these databases were really at the forefront of what they were trying to do when they were founded and have had updates since, to be perfectly honest, as a bioinformatician, they could be a little bit more computationally advanced here in this year of our Lord 2025. They don't have all the features that we would want to integrate into our pipelines and our analyses. And this is really the challenges that we are trying to solve with Pathoplexus. I don't have time to go into them all today during this short talk. I'm happy, of course, more than happy to talk about them. But in particular, we are aiming to be easy to use. That means both to upload the data to and to get the data back out to use in your analyses. We also have very clear data use terms that I'll cover more in a minute, but that outline how data can be used and how it shouldn't be used. In addition, we think that data versioning is critical. We need to be able to fix mistakes. We are not perfect. We make better versions of sequences. We get our dates mixed up. When we make those changes, we should be able to see clearly what it was in the past and how it's changed between different versions. And then finally, and perhaps most importantly, we are absolutely dedicated to transparent governance and being part of the community that we serve. As I already mentioned this morning, Pathoplexus currently supports 9 pathogens, and I'm really proud of that because that's up from the 4 that we launched with. So we currently support Crimean Congo hemorrhagic fever, Ebola Sudan, Ebola Zaire, HMPV, measles that we just added this August, Mpox, RSV A and B, and West Nile virus. And we're focusing on viruses that we feel, as I said this morning, don't have a home, those where we can make a difference by adding them to Pathoplexus, making that easier for the community. And we don't do this top-down, we do this in association with communities. So communities actually submit to us that they are interested in having their pathogen in Pathoplexus. Our executive board takes that into account and votes on whether to add a pathogen. Because we don't want to be running into other people's associations, their collaborations they're working with on different viruses and telling them they need to be on Pathoplexus. We want that to be something that's genuinely useful from them and powered by the users. I want to talk a little bit more about how data sharing and our data use terms works. As I've already mentioned, we've aimed to make this as easy as possible. So you can upload data to Pathoplexus by simply dragging and dropping a FASTA file and a metadata file into our website. That's it. You can even map the columns so you don't have to use our column names. If that even sounds a little bit too much, you can kind of see there's a second tab there that's a single sequence submission. You literally just fill in the fields that you feel like you're able to share and copy and paste the FASTA at the bottom. So even someone who's not comfortable with FASTA files or something can still share data. On the other end of the spectrum, we also have an API. So if you have a sequencing pipeline, you don't need humans clicking web pages. You can have your pipeline submit your sequences automatically. So we're really trying to cater to all of the user needs and abilities. Now, once you share that data, you have 2 options in how to share it, which I touched on briefly this morning. First, you can share that data openly, which is essentially the same as putting it on ENA or GenBank, and indeed, that's exactly what happens. So this means that anyone can use your data. They don't need to get in touch with you. What's different is that you can upload it to us this really easy way, dragging and dropping your files, and then we will put it onto INSDC for you. So we will take care of what for many people can be the hard part. We are a broker with ENA. What that means is that this will still be your sequence. You will be the authors. It will come from your institution. We can even add it to your biosample if that's what you like. So it's very much your sequence. We just do the wheel greasing part of getting it up into ENA. On the other hand, if you're worried about someone scooping your data, publishing on it before you can, but you still want to share it quickly for public health, you can choose to share your sequences as restricted use data. I want to really emphasize that's restricted use, not restricted access. All of the data on Pathoplexus is open and available. However, with restricted use, it cannot be used for everything. In particular, restricted use data cannot be used in publications and preprints without the explicit consent of the submitter. And when we say that, we mean that. It's not I emailed them 6 times and they didn't reply, so I did it anyway. They either need to be an author on the paper with you, or they need to send you a letter saying that they didn't want to be an author, but it's okay to use the sequences. Otherwise, you can't use the data. However, this is time limited, so you can have that data be restricted use for up to 1 year. It can be less if you want, and you can always release it early. That's absolutely fine if your paper comes out, for example. But otherwise, after 1 year, the data will become open. And this we see as a kind of balance. We think it's absolutely critical that people do have protection so that they feel like they have time to do an analysis and get a publication out while knowing no one else can publish. But we also know that long term, it is the most valuable to have data that can be used by others, because that is how we, for example, repeat and show that the same analysis can be done. Replication is at the heart of science. And also how we build on that data. If we have new variants next year, we need to be able to see how they compare from the variants from the year before. So we do think that having that data long term in a stable repository that's open is a good solution and balance here. Another thing I really want to emphasize is that Pathoplexus is designed to be part of this ecosystem, and it was fantastic to be able to talk about this a little bit this morning, because we're not trying to be a new island or a new castle, very much the opposite. We want to fit and be part of something that's bigger. So I've already talked about how data from Pathoplexus always ends up eventually on INSDC, so ENA and GenBank. We also do pull data into Pathoplexus from INSDC as well. This means that in Pathoplexus, you can access all of the same data that you'd get on INSDC, plus the restricted use data. So it's a very convenient way to do your analysis. Or even if you're just looking to use the open data, maybe you just find it more convenient to access that through Pathoplexus. I want to be really clear, though, we never hide that that's where we got the data from. It's always incredibly clear that it came from INSDC. You can always see the original accession number, and you can always link back to it on ENA or on GenBank. That's always possible. We do this just as a way of making it easier to get a hold of all the data you might be interested in. In addition, we wanted to make sure that, of course, we're empowering research. Now, all of the databases that I've talked about today support research and public health. That's why we have them as a database. But we also recognize at Pathoplexus that even though publications are still critical, they're not the only way we do science anymore. I'm sure many of you, like me, used other tools in the pandemic that were also critical. So you may have used analytic tools that were developed for SARS-CoV-2 that were enabled you to do very specific things. Or you may have used one of the many websites that developed to track cases, to track variants, to explain different mutations. These are critical for public health and for science communication, and we need to make sure that we're able to build on those tools because they empower not only public health interventions and informing the public, but of course further research as well. So at Pathoplexus, we have built a comprehensive API so that we can build these tools on top of that data very easily. So it can be done. People can make new analyses and new websites that build on top of that data. And one thing that makes us stand out a little bit is that at Pathoplexus, we do actively enable that the data can be redistributed. Now, of course, there's rules on this. You need to take the data use terms with you because it needs to stay attached to that restricted use data so that its protection stays. But at Pathoplexus, we don't see this as like, we can have all the data, it's ours, and you can't do anything about it. This is not our data, it's your data, it's the community's data. We are holding it and we are doing our best to be a good steward of that data and to enable good research. But if the day comes when we are not that good steward anymore, it's critically important that someone else could take that data, preserve the protection of the sequences, but put it somewhere else that the community perhaps prefers, so that data isn't trapped and so that no one is kind of held to ransom by Pathoplexus. In addition, as I mentioned, we also think it's really important to have versioning and community input into these sequences. So for example, if a submitter sees a mistake in their sequence, they can always update it, they can fix a date, or even have a new sequencing run that's maybe a little bit more accurate, and they can upload that and you'll see it as a version change. In addition, if members of the community spot a change, we can either contact the original submitter if it's a direct upload so that we can show them that maybe there was a mistake. We can even actually queue up the correction so that if they agree with us, all they have to do is hit agree and that correction will go live, which can be a lifesaver in an outbreak when you're kind of too busy to do a lot of corrections. Or if that data came from INSDC, we can actually make that correction ourselves, which can be a huge help because otherwise we all have to make that date correction or that country correction in every single individual analysis on everyone's computer. So let's fix it in one place so that we can benefit from that more accurate data. I also want to touch on as I said, our commitment to transparent governance and our commitment to being part of the community. We have an amazing board and membership. Pathoplexus is a separate legal association. We're not associated with the university. And that was really important to us because we want to be sure that it doesn't have to be attached to one person forever. Everyone in Pathoplexus could eventually rotate out. New people can come in, and Pathoplexus remains independent and a separate organization. We have a wonderful executive board, highlighted at the top, 5 people from 5 different countries, 5 different continents, my apologies, and I'm not just saying that because I'm on it, I swear. We also have an incredibly diverse set of members from over 10 countries and all sorts of expertise ranging from bioinformatics to virology to even legal ethics. And these members have real power. If the members don't like what we're doing, they can vote out the executive board and vote in a new one. So this holds us accountable, and it makes sure that we have an incentive to make sure that we are doing things that are empowering the community and that are what the community wants. And we recognize that may change over time, and that's good, and we should be able to respond to that, and we should be incentivized to respond to the community's changing needs. We also are committed to transparency, so all of our minutes and all of our resolutions are available online on our website. And we have, of course, monthly meetings with the PHAGE community, so we are part of Working Group 6. We meet once a month. Please join us if you're interested. And this allows us to have an incredibly valuable way of finding out what do people want, what are suggestions, maybe a problem or a feature that's missing. So this is an excellent way for us to stay in touch with those who are using Pathoplexus. I want to just highlight, even though we've really only been running a year, we are so honored to have been able to play a role in outbreak response already. So for example, both for the earlier outbreak, Ebola outbreak in Uganda at the very beginning of this year, and as Placide talked about this morning, the more recent Ebola outbreak in DRC, both of these outbreaks chose Pathoplexus as the place to share their data. So not only did they do an incredible job of sharing that quickly, they trusted us with sharing that data as restricted use as soon as they had those sequences. In addition, we are the database that had received the most mpox sequences in 2025, and many of these also come to us as restricted use. And to me, this really highlights that there is a need for different ways of sharing data in the community. People are seeing that this fills a gap, and in particular, they are seeing that having these time-limited protections enables them to be able to share faster while still knowing that a publication can follow. And I'm really honored that these people have chosen to share this data with us, because I think it does show that we can encourage data sharing by creating an environment where people feel safe to share. One of the last things I want to touch on is that we're also very aware that, of course, data sharing during kind of peace times is very different from the data sharing that we might need if we have another, or if and when we have another pandemic. And Pathoplexus, we're really flexible. We have designed this so that we can respond rapidly. So within minutes, we could add a new virus and have it live to receive submissions in, for example, a new pandemic scenario where that was critical. It might be fairly basic at the start because we may not know much about a new virus. That's kind of how novel viruses are. But what's also good is that as we learn more, as we get annotations, as we get reference genomes, clades, these kinds of things for the virus, we can add those and they will retrospectively be applied for the samples. But what's critical is you can share that sample as soon as you have it. We can start that precedent early with a new virus, and indeed making that restricted use if that's what people feel like they need to share that sequence. I don't have enough time today to talk about the code behind Pathoplexus, but I want to mention it briefly because it's also really important. So we've also built the code that runs the database of Pathoplexus. It's called Loculus. It is, of course, open source and transparent, like everything that we do. It's also highly flexible and pathogen agnostic. And so we're really hopeful to develop Loculus into another kind of data sharing solution, where this could be, for example, something you could use to store sequences at your lab, at your university, or indeed even at your country level. And what we'd like to see is, of course, have that be easy ways to then share from your local Loculus up to Pathoplexus, or even to different Loculus instances, perhaps your collaborators. This is something that we're still working on, having these local releases be something that we're really able to say to the community we're ready to use, but it's something that we'll be focusing on in 2026, so please stay tuned for more news on that. So finally, I really hope that today I've been able to give you this brief introduction into something that we are so excited that we've been able to bring to the community. A data sharing solution that helps to empower sharing, to make sharing faster, easier, and hopefully also more equitable. And of course, this then enables all of those downstream things, enabling prospective pathogen research to keep us safer from what may come tomorrow, and also enabling better surveillance so that we have a better idea of what's out there and how to reduce that health burden. My talk is really getting ahead of me. Finally, if you have any questions, there are many Pathoplexus members here. There are many Working Group 6 members here. Please come and find me. I will be happy to chat. You can also talk to my Working Group 6 co-chair, Arthur, or the Pathoplexus co-founder, Karan, who's also here. And we have wonderful stickers. So if that's what you need to come and speak to us, please come and ask for a sticker, and we're more than happy to talk to you and answer your questions. Thank you so much.